Welcome back to Cell Biology. We are in the middle of our chapter discussing how cancer forms. We were just discussing how things like density independence, anchorage independence, being able to keep telomerase activity, how those are able to contribute to a cancer growing, to being able to divide. But it's not what actually causes the cell division in the first place. What usually causes a normal cell to divide in the first place is a growth factor. So your body has sent the signal that it is time for the cell to divide. A cancer cell is not going to wait for a growth factor. A cancer cell is going to create its own signal to divide. It might create a new receptor on the cell or a deformed receptor on the cell that is constantly saying, hey, getting a growth signal, even when it's not. It might start making its own growth signal, so it's constantly getting a growth signal from itself instead of how it should be. There are different ways that cancer cells are able to grow even when there's not a healthy growing signal. There's also disruption of cell cycle controls. So things that are normally going to tell the cell, hey, let's stop and check that this is fixed before we move on. Those cell cycle controls are not there. So the cell is just constantly going through that cell cycle without any braking system. That's also going to contribute to the cancer cells proliferating. So one of those points in the cell cycle that normally triggers the cell to pause is the restriction point. If the normal cells are growing under suboptimal conditions, so it doesn't have a growth factor, it doesn't have ideal levels of energy sources or nutrients, it's going to stop growing at that restriction point. That is what is going to stop the cell from going from G1 into S. A normal cell is going to say, we don't have these good resources available, so we're not going to invest in copying our DNA and dividing. It just kind of pauses at that restriction point. Cancer cells don't pause at the restriction point. They don't respond appropriately to those external signals. They don't recognize that they don't have great nutrients around. They just keep growing. They don't respond to internal conditions that say, hey, we need to stop and check our DNA before we divide. Hey, we need to stop and do something before we divide. They're just going to keep going, just keep dividing. For some cancers, growth is due to a failure to undergo apoptosis. There's a word we've defined already. That is controlled cell death, where the cell is going to realize that it is no longer functioning correctly or it is no longer contributing to the health of that organism, and it will signal for that cell to go through apoptosis or cell death. For some cancers, they don't properly go through apoptosis when they should. So they're going to be growing and continuing to exist and grow more when they shouldn't be. They have different pathways that we're going to talk about in our next chapter about how they're able to kind of stop those apoptosis signals. So cancer development really requires multiple steps. There is first this initiation, when the cell has become precancerous, we would say. It is precancerous. It is sensitive to developing further mutations, but at that stage, it's not really dangerous. Then it's going to, over time, pick up more and more mutations as this cancerous cell is continually responding to maybe some carcinogens or sunshine, UV light. Something is going to continue to cause mutations in these cells. Over time, that tumor is going to keep growing, and it can differentiate into different kinds of cells. You might have some that grow more, and so they are selected for more epigenetic changes, more proliferation. That is tumor progression, as you are developing more and more unhealthy, abnormal cells. If it's a benign tumor at its original location, it's usually going to be pretty easy to remove. But one dangerous thing about tumors is if their uncontrolled proliferation is coupled with the ability to spread and start causing havoc through the entire organism. Around 90% of cancer deaths are due to that spread of the tumor. It's not just having extra cells growing in a ball, it's having extra cells going through your body, doing things they shouldn't be. One thing that allows a tumor to grow is a process called angiogenesis. Angiogenesis is the process of growing new blood vessels. And if a tumor cannot grow new blood vessels into itself, it is not going to be able to get nutrients to grow. In 1971, a scientist named Judah Folkman did some experiments seeing how signaling molecules could trigger angiogenesis and how growing new blood vessels was related to a tumor's ability to grow. So he took a thyroid from a rabbit and grew it in a glass chamber away from the rest of the organism. It is isolated here. There are a few cancer cells that were injected into the gland, and there were plenty of nutrients to keep it alive. And those tumors did grow for a couple of days. But once they got to about 1 millimeter or 2 millimeters big, they stopped dividing. They didn't have those blood vessels growing into them, and they were not able to keep growing. However, when those tumor cells were taken out of the thyroid and put into an animal, they started dividing again, and they divided into large tumors. So the difference here is in the isolated gland in that little glass bottle, the tumors couldn't link up with blood vessels, so they didn't have any way to get what they needed to grow. But once they were in the animal, the tumors were able to grow blood vessels. They got the blood vessels in the animal to actually reconstruct themselves into the tumor, which allowed the tumor to grow larger. We can see similar results with tumors in rabbit eyes. So here's an example of the eye. If it's here in this anterior region, the front region of the cell, the front region of the eye, where there's not a lot of blood supply, it stays pretty tiny. See how small that little green dot is? So here's the line showing you how big that tumor is. But if it grows closer to the iris, where it has access to a blood supply, so it starts having these little blood vessels growing into it, and this green spot is much larger than this one. Once it has access to that blood vessel, it can grow larger. So angiogenesis, or the ability to grow new blood vessels, is required for tumors to grow beyond just this couple of little millimeters big. Cancer cells have been shown experimentally to make molecules, to release molecules that activate angiogenesis. Two of these molecules are VEGF, or vascular endothelial growth factor, and FGF, or fibroblast growth factor. These are two molecules that a tumor can make that activate angiogenesis. So when a tumor starts producing these two molecules, it is going to cause the blood vessels around that tumor to remodel into the tumor. When the tumor is releasing VEGF and FGF, it gets released from the tumor into the surrounding tissue, and it's going to go to the endothelial cells on blood vessels. So these are the cells that are lining the inside of the blood vessels. When those endothelial cells pick up those growth factor signals, they think, oh, it's time to start growing. So they start growing. And when they grow, they release something called MMPs. These are matrix metalloproteinases. MMPs are really good at breaking down the extracellular matrix, which kind of functions as a fence or a gate around the cells. When that extracellular matrix is broken down, you no longer have that gate, so endothelial cells are able to migrate into the surrounding tissues. They no longer have any sort of boundary keeping them out. And when the endothelial cells start going into those tissues, they're going to start organizing themselves into little tubes. Those little tubes are going to become the capillaries and blood vessels. However, it's not enough to have VEGF and FGF there. Your cells are also going to have angiogenesis inhibitors that they make to protect themselves from inappropriate blood vessel formation. So VEGF and FGF not only have to signal to grow, but they also have to overcome those inhibitors. Some of those inhibitors of angiogenesis are angiostatin, endostatin, and thrombospondin. So a tumor that's able to trigger angiogenesis is both increasing angiogenesis activators like the VEGF and the FGF, and decreasing the angiogenesis inhibitors like angiostatin, endostatin, and thrombospondin. Then, now that this tumor has a blood vessel growing into it, that blood vessel is not only going to bring it nutrients, but also give it a ticket to spread through the body. So the ability to metastasize starts with angiogenesis, and then these tumor cells are able to travel through the tissues, get into the bloodstream. Then that bloodstream goes through the whole body, so they can just kind of ride the rails until they find a spot that they want to get off. Then they can leave the bloodstream, and now you have cancer cells in a new area. So this could be breast cancer tissue. These cells are leaving the breast cancer, traveling through, and suddenly now they are growing on your colon, or growing on your stomach, or growing on bone, and going to cause problems wherever they're growing. So the first step in metastasis is invasion. It had to invade those tissues. It had to invade those vessels. One protein that is downregulated in a motile cell is E-cadherin. So there are these cell-to-cell adhesion proteins that keep your cells connected to each other to form tissues. And these cells that are connected to each other, in a cancer, they're going to stop being connected. So they're often missing or defective in these cell-cell adhesion proteins. One example would be E-cadherin is often missing or defective in cancer cells. And if you are able to restore E-cadherin expression in the cancer cells, it inhibits its ability to invade. So as you increase the expression of E-cadherin, you increase the ability of the cells to adhere and stay in the tissues where they're supposed to be. You reduce the ability of that tumor to metastasize if it is attaching to other cells in a normal pattern. Okay, so it has to be able to attach or detach. Another factor in invasion is motility. Motility is the ability to move. This is going to be stimulated by signaling molecules. Surrounding tissues can stimulate these signaling molecules. The cancer cells themselves can originate with these signaling molecules that are going to act as chemoattractants, or chemicals that attract the cells toward them. So your cancer cells start moving towards a chemoattractant. Rho family GTPases plays a role in this. That's something you may be interested in looking up. We don't have time to talk about that within the context of this class today. Cells also secrete something called proteases. Proteases are going to break down those protein structures that normally form a barrier, form that fence around regions where the cells shouldn't be spreading. A couple of barriers would include the basal lamina. That basal lamina that is underneath a lot of tissues, that is a barrier that the basal lamina has to be broken down. Or the extracellular matrix would have to be broken down for the cells to be able to travel outside of their area. So cancer cells will secrete proteases that degrade the basal lamina. They can also secrete plasminogen activator, which is going to turn plasminogen into plasma and activate it. And that's what will break down the basal lamina and the extracellular matrix. However, when cells are traveling through the bloodstream, that's a pretty harsh environment. If they're able to survive, they can ride those blood vessels throughout the body. They can even enter a lymph node and ride that throughout the body. But that ride through the bloodstream typically is pretty inhospitable. So at this point, we're going to talk about what happens to a cell once it's been metastasizing. What are some of those genetic underpinnings that lead a cell to become metastatic or cancerous? I'll see you back in our next video when we talk about that. Bye.